So far in this series, we have gone through the most common creationist talking points pertaining to cosmology, planetary science, abiogenesis, evolutionary biology, and paleontology. Let's continue by zooming into the molecular level so that we can discuss evolution from the standpoint of genetics and molecular biology. Most creationists do not comprehend genetics on even a high school level. For this reason, it is very easy to run circles around less educated creationists within this unintuitive sphere. If they don't understand what DNA is or what it does, it's like shooting fish in a barrel. More sophisticated, intelligent design proponents focus most of their propaganda on this area as well, because it is so much more esoteric, and lying with confidence about things that their targets don't understand gives them a high probability of success. There's lots to discuss here, so let's get started. First, we need to go over some background information. Creationists don't understand genetics, but you should, so let's hit the basics in ultra-abbreviated fashion. As always, I have tutorials on biochemistry and biology that explain everything we will discuss in greater detail, so head there after this series if you want to take the next step. For our purposes, let's make sure we know what DNA is. DNA is a nucleic acid, which means it is a polymer of nucleotides. There are 4 different nucleotides in DNA, each made of a sugar, a phosphate, and a nitrogenous base. These bases are abbreviated as A, T, C, and G. Nucleotides come together in different combinations to make DNA, which is made of 2 strands that coil together in a double helix, with the bases pointing towards each other in the interior. The way the 2 strands anneal is according to base pairing. A pairs with T and C with G. Every organism has a genome, which is made of some number of chromosomes, which are very long DNA molecules with billions of base pairs wrapped around proteins called histones. These chromosomes are the genetic code, which contains instructions for the assembly of the organism during embryonic development and its maintenance throughout its life. This occurs through transcription and translation. In transcription, small portions of the chromosome called genes are opened up by enzymes in order to generate an mRNA transcript. This occurs by the same rules of base pairing as for DNA, but mRNA is a little different. Then the mRNA is translated by a ribosome, again through simple base pairing, in order to generate a protein. This happens because the tRNA molecules that match up with the mRNA transcript, 3 bases at a time, are attached to specific amino acids, and the amino acids join together to form a polypeptide, which folds into a protein. Proteins do almost everything in your body. There are structural proteins, there are enzymes that do chemistry, there are receptor proteins that receive messages and trigger physiological functions, and so forth. One of the most important aspects of genetics for our purposes here is the way that the genome guides embryonic development to yield a particular type of animal. Unfortunately, this is exceedingly complex, but we can at least describe Hox genes, which are a group of related genes that specify regions of the body plan along the head-tail axis of an animal. Encoded in these genes is the number and positions of limbs and other structures that will develop along the body. This is because the genes code for transcription factors, which are proteins that help turn other genes on and off by selectively binding to them, thus either promoting or inhibiting gene expression at hundreds of other sites on the genome. Because Hox genes have products that regulate so many other genes, changes to these genes can have dramatic effects on the developing organism's body plan. With that covered, we now have to discuss mutations. Mutations describe changes to the genome. There are many different kinds of mutations, and they can occur for many different reasons. The most simple mutations are point mutations. That is a change in just one base pair, from whichever base it was supposed to be to some other base. This can occur due to endogenous or exogenous mutagens, carcinogens, UV light, or totally natural decay processes where radioactive nuclides within the body decay and produce a high-energy particle that strikes a base in the genome of a particular cell. Errors can also occur during replication, where the enzyme creating the new strand makes a mistake. Repair enzymes exist to fix these errors, as well as the other mutations we described, but they don't catch them all, so point mutations happen in every organism all the time. Most are neutral, many of the rest are harmful, and some are beneficial. It depends on whether any of the amino acids in the resulting protein change, and how that affects the function of that protein. If it's an enzyme, it could function more efficiently, or less, or it could lose function, or even change functions if several mutations accumulate. We have to understand some other types of mutations that can occur. There are mutations like insertions and deletions that change the reading frame of the gene, which usually result in a non-functional protein. But there are also macromutations. Very significant changes to the genome can sometimes occur. For example, gene duplication. During replication, the enzyme can copy a section of DNA more than once. Additionally, during crossing over between homologous chromosomes in meiosis, a type of cell division, misalignment can result in two copies of the same gene getting into one chromosome. With two copies of the same gene, neofunctionalization is now possible. One copy of the gene can be retained, while the other gene can slowly accumulate mutations and therefore yield a totally different protein product. No function is lost, while a new one can be gained, and studies show that around 50% of gene duplications lead to functional divergence. A similar situation occurs with orphan genes, or de novo genes. There are sections of DNA called promoters, which sit next to the coding region of a gene that an enzyme will interact with, resulting in transcription of the gene. A mutation event can sometimes rearrange sections of the chromosome, which result in a promoter being placed next to a previously non-coding section of DNA. This results in a brand new gene instantly with a totally random sequence, and therefore a totally random protein product. This protein might be useless, but it might not, and any modest functionality can be subsequently molded through natural selection. The potentiality of a process like gene duplication is magnified tremendously when entire chromosomes duplicate. This is again typically due to errors during cell division, where homologous chromosomes or sister chromatids don't separate during meiosis, so one cell ends up with multiple copies of the same chromosome. This leads to the possibility of many different genes going through neofunctionalization, which can have dramatic effects for an organism. Now, if any of these mutations occur in the somatic cells of an organism, the effect is usually minor, but if they occur in the germline cells of the organism, or the cells that produce gametes, meaning sperm and eggs, then we are looking at the offspring having a significantly different genotype than the parents, and thus potentially a different phenotype, which refers to actual morphological differences. These are typically very tiny, but sometimes not so tiny. It simply depends on how those proteins influence embryonic development and the determination of the body plan. We have physically observed instances where even single point mutations can have drastic effects on an animal's morphology, like legs growing from a fly's head instead of antennae, extra pairs of wings, and other major changes. The implications for evolution are obvious. With this understood, we can now begin to outline the various mechanisms by which evolution operates. Whereas Darwin envisioned only natural selection, over the 150 years since, we now know that there are many other mechanisms also at play. Apart from mutation, which produces genetic variety, and natural selection, where organisms better suited for survival and reproduction have their genes more likely to proliferate, there are a few other important ones. Genetic drift describes a change in the relative frequency of an allele in a population due to chance and random sampling. An allele is a copy of a gene. Most animals have two copies of every gene, one from each parent, and each copy is called an allele. Alleles can be dominant or recessive, and this genotype can dictate the phenotype, or the version of a particular trait that is expressed on the organism. There is also gene flow, which describes the movement of genes from one population to another, often due to migration, which can increase genetic diversity. Then there is sexual selection, or non-random mating based on perceived evolutionary fitness. This operates by criteria that are quite different from survivability, and can produce interesting morphological features. Horizontal gene transfer, niche construction, epigenetics, the list is long and we need not dwell on every detail for our purposes. As we can see, evolution relies largely on population dynamics and changes that occur on the genetic level, which Darwin could never have understood in his time, before we even knew that DNA existed. Creationists relentlessly talk about Darwinism and Darwinian evolution, and in general never shut up about Darwin for this specific reason. He lived a long time ago before we actually understood evolution, so they will try to present evolution in this primitive lens. Young Earth creationists and intelligent design advocates alike will always return to the same mantra: random mutations and natural selection are insufficient in producing the variety of life. They will all say this every chance they get, and yet, even with the couple of minutes of background information we just went over, we can immediately see how dishonest this is. When they say random mutation, they mean point mutations. These are more limited in scope. They are ignoring macro mutations like gene duplication, de novo genes, or chromosome duplication, which produce significant discrepancies in the genome in a single generation. And when they say natural selection, they are limiting evolution to a singular mechanism, ignoring the others we described. This is a deliberate misrepresentation of the scope and potentiality of evolutionary processes. More detailed attempts to misrepresent genetics will take advantage of the negative connotation of the word mutation to pretend that all mutations are harmful. They will say things like, mutations degrade genetic information, as though a bucket of toxic green goo has fallen on the DNA and is giving it chemical burns. In reality, mutations are simply changes to the genetic code. As we said, with point mutations, the vast majority are neutral. The mutation does not result in any change whatsoever to the protein product. Of the remaining possibilities, some are harmful and some are beneficial. We have to look at the change made to the protein. Is the amino acid that changed in the active site of an enzyme? Does this cause it to bind to its substrate more tightly, or not at all? Any of these possibilities could be bad or good, depending on the situation. Significantly harmful mutations are not often observed because those organisms tend to die quickly without reproducing, so we disproportionately observe beneficial mutations and improved function, which can appear like design to the unprepared mind. Another creationist fixation has to do with probability. They will take any enzyme, like say DNA polymerase, and remark how unlikely it is for that to have arisen by chance. In order to make it seem impossible, they will play a devious numbers game. They'll say, here's an enzyme with 300 amino acid residues. There are 20 amino acids, so the probability of this precise sequence evolving by chance is 1 in 20 to the 300th power. Their targets will gasp at the infinitesimal odds and conclude that it can only exist because God willed it so. This is ridiculous. That precise sequence is far from the only sequence that will perform that function. Many of the amino acids in the active site of an enzyme have limited variability, although even those have some, but the rest of the amino acids contribute only to the overall shape of the enzyme, and thus have tremendous variability. There are sets of amino acids that are extremely similar. Amino acids have the same general structure, plus a variable side chain. Here are a few that have an alkyl side chain, meaning they consist of only carbon and hydrogen. Anywhere we find one of these, we could easily swap to another similar one, and the effect on the enzyme would be practically non-existent. Only if we switched from something like a hydrophobic to a hydrophilic side chain, like alkyl to one containing a hydroxyl group, could that potentially have an impact on the folding pattern, and even then, it is unlikely to be significant. So we are saying that the majority of the amino acid residues could be completely different, and the function would be identical or similar. Furthermore, new enzymes don't have to function optimally. They can perform a function with moderate efficiency and then get refined by natural selection over time. And finally, we know for a fact that many different sequences can perform the same function because different organisms have different versions of the same enzymes. Looking again at DNA polymerase, different organisms have different polymerase enzymes. They have different sequences, and we give them different names, yet they all copy DNA. With genetic engineering, we manipulate the amino acid sequence of enzymes to make them perform more efficiently than the natural counterpart. There is tremendous variability and nothing is perfect in the first place, so this is lazy propaganda on the part of creationists. Nature creates many sequences, stumbles upon functional sequences by chance, and those are optimized and amplified by natural selection. Plenty of research has been done which corroborates the fact that totally random sequences are an abundant source of bioactive RNA and peptides. This is not controversial. Recalling the creationist claim that microevolution is possible but not macroevolution, this seems even more ridiculous in the light of molecular biology. There is no mechanistic distinction between the two. It's all based on mutations. We already explained how certain point mutations, and especially macromutations, can produce significant morphological change, even in a single generation. Changes to Hox and tail genes can dramatically revise the body plan. Differential gene activation can affect cell differentiation, thus allowing cells in different regions of the body to do different things. And in general, all of this hinges on creationist incredulity regarding embryonic development. Creationists have no idea how this process works. If a creationist tells you that evolution can't produce large-scale changes to a body plan, ask them how a body plan is implemented during embryonic development in the first place. Just ask them point blank. All animals begin as one cell, right? So how does that become a specific animal with a specific body plan? Just sit and wait for an answer. If they offer anything at all, it will be vague, like the DNA has the instructions and that's how you get the body plan. But how? Ask specifically and repeatedly. How, mechanistically, on the molecular level, does the body plan develop? I bet you $1 million that not a single creationist will give you a detailed answer. The reaction to this is therefore, you have no idea how the body plan arises. You have no idea what happens on the cellular or molecular level as a single cell becomes a complete organism. Yet you are somehow an expert on what this process can and can't produce? Hopefully this will embarrass them enough to do some learning, or at least question their beliefs. If they are quiet enough to receive more information, you can tell them about how body plans arise from the time-regulated expression of specific genes. So mutations in those genes can change the body plan. Tell them about all the kinds of macromutations they definitely have never heard of. Tell them about examples of visible change that result from even point mutations, like flies growing legs instead of antennae. Creationists do not know these things. There is no barrier preventing new species from emerging. Mutations accumulate and the organisms change accordingly, so there is no meaningful distinction between micro and macro evolution. Getting more specific on the anatomical level, looking at certain transitions, like from sea to land or from land to sea, is it really that shocking to go from hands and feet to webbed hands and feet to fins? Is it really that hard to wrap one's head around how that might occur? What are the necessary changes to the genome that can push animal morphology in that direction? Even with humans, early in embryonic development, hands and feet form in flipper-like paddle shapes. Fingers and toes subsequently separate, elongate, and then lose webbing that is between them. This loss of webbing is due to interdigital necrosis, a type of cell death, so genes are selectively expressed in those cells that result in those cells dying, such that the fingers can separate. If those genes are not expressed, the webbing can remain, and there are disorders in humans where precisely this happens. If this occurred with animals in aquatic habitats, it would be an enormous fitness benefit. It's a heritable trait, so all of a sudden many organisms within a population can have webbed extremities. Further modification to the genome can affect embryonic limb development such that a more fin-like structure is retained, and then you have fins. All of this is determined during embryonic development, where the limbs are going through different stages anyway. Large-scale morphological change can occur simply by silencing a certain aspect of embryonic development in a particular part of the body. It's as simple as blocking a single gene from being expressed. And by looking at this example of webbed extremities, we are even identifying intermediary structures. Life wouldn't have to go straight to fins. There can be many gradations in between, such as hybrid structures that are not optimized for either walking or swimming. Even this kind of anatomy can be considered useful because such an organism can suddenly evade predators in either habitat, land or sea. Continuing with embryonic development, a favorite creationist talking point is to reference the early history of embryology. In the 19th century, Ernst Haeckel pioneered this field by observing the embryos of many different species and hand-drawing their development at various stages. Comparing these developmental pathways offers many clues regarding evolutionary relationships, since many aspects of development are shared between distantly related animals. For example, all chordates, including mammals, birds, reptiles, amphibians, fish, tunicates, and lancelets, have a notochord, dorsal hollow nerve cord, pharyngeal arches, a postanal tail, and an endostyle or thyroid gland at some point in their development. Some chordates have all of these characteristics as adults, like lancelets, but others lose at least some of them later on. For example, humans lack an intact notochord, pharyngeal arches, and postanal tail. Our notochord gets broken up to become the intervertebral discs. The pharyngeal arches become parts of our face and neck, and the postanal tail shrinks away to a tiny vestige of its former self. But these structures still do form during embryonic development. We have 10 to 12 tail vertebrae before they retract into the soft tissue. It would seem odd from the perspective of design that embryos would go through stages where structures grow that will not be present on the mature organism. But it makes perfect sense in the context of evolution, where development begins a certain way, and then mutations cause gene expression to change at a particular stage, where existing structures then become reduced or modified. What Haeckel correctly showed was that all chordates go through a phylotypic or pharyngula stage, where the embryos of animals as diverse as elephants and sharks look extremely similar. What he got wrong was an idea he referred to as ontogeny recapitulates phylogeny, or the idea that organisms recapitulate their evolutionary ancestry during development. There is some truth to this notion, like the way that mammalian ear bones detach from the jaw during development, but broadly speaking, the idea is not accurate. By this, we mean that a mammal doesn't go through a specific fish stage, followed by an amphibian stage, and then a reptile stage, and so forth. Later embryologists recognized the extreme similarity early on, but that embryos then diverge radically in morphology. The drama enters when people accuse Haeckel of fraudulence. While this is primarily creationists, it also included a few biologists. They will say that he fudged the scale or exaggerated characteristics, since they were hand drawings. In reality, he made them the same size for ease of comparison. Additionally, he accidentally used the same drawing for 3 embryos in his first edition, but this was pointed out to him and he fixed the images for later editions. Creationists essentially make a lot of fuss out of nothing, and when they bring this up, you can ask them to clarify what they mean and where they got their information. They will have no response. Even more important, however, is the fact that Haeckel was undeniably correct. Embryos do go through these stages, and various mammalian embryos are virtually indistinguishable for a significant portion of development, something that is easily confirmed in the modern era, where we no longer have to draw embryos. We can easily get real-time images of them. They look more or less just like Haeckel's drawings. Moving on, there are some classic arguments from intelligent design proponents that must be neutralized. The first is the so-called waiting time problem. This one says that adaptations which require multiple mutations are impossible because of the improbability of those specific mutations occurring in a reasonable time span. There are a lot of issues here. First, nature is never going for a specific set of mutations. Mutations occur, and they are either selected for or they aren't. And these mutations do not have to occur in a specific sequence. They could occur in any sequence across an entire population and then come together during the recombination phase of sexual reproduction, such that each parent can have one mutation and the offspring has both. Furthermore, the idea that each individual mutation can't be selected for because only the entire set confers the beneficial adaptation is incorrect. Individual mutations could still confer a slight survival advantage and therefore proliferate statistically. A similar argument from ID proponents is that of irreducible complexity. This states that some biological structures are so complex, with so many interconnected parts, that it can't possibly have evolved, because removing just one component renders the structure unusable, and therefore none of the individual components can have been selected for, such that it could have arisen piecemeal. It's all or nothing. Their favorite example of such a structure is the bacterial flagellum. They will describe this structure using as much mechanical language as possible, drawing parallels to actual human machinery. This is part of the deception. Humans built these things, so God must have built this thing. In actuality, this is a facade. First of all, when describing something like the bacterial flagellum, they are never honest about how many different types of bacterial flagella there are and the wide range of complexity they display, which demonstrate the range of solutions nature can invent to solve the same problem. Just because a flagellum is highly complex now, it doesn't mean it was always that way. Individual components can have complexified after the general function was reached, and there are extant flagella which lack components that others possess, yet they still function. Second, they always ignore the concept of exaptation. Instead of waiting for entirely new proteins to arise, like those that become the components of a bacterial flagellum, existing proteins can be slightly modified to develop entirely new functions. Sometimes a very small number of mutations is enough to achieve this, and a new protein suddenly exists for inclusion in some apparatus. Third, any such irreducible structures are never actually irreducible, and oftentimes reduced structures specifically do exist. For the bacterial flagellum, a corresponding reduced structure is called the injectosome. This is a needle-like protein complex that uses components of the flagellar apparatus, but instead of generating motion, it secretes effector proteins. It doesn't matter whether the flagellum evolved from the injectosome or vice versa. The claim that ID proponents make is that with any irreducibly complex apparatus, there is no possibility of any reduced structure. Yet, here we see one. It uses only some of the components of the flagellum and serves a totally different function. We can say the same thing about absolutely any molecular or anatomical feature that they claim is irreducibly complex. When they bring up eyesight, they ignore everything we know about the evolution of eyes and the many types of eyes in the animal kingdom, ranging from a tiny bundle of photoreceptors to complex eyeballs like those in humans or squids, and everything in between. In fact, it is known that eyes have evolved independently dozens of times in different species, as an example of convergent evolution. Vision is a highly valuable trait, so any features that bestow this function will be heavily selected for, which is why it happened so many times, resulting in many different types of eyes. It's not necessary for entire eyes to suddenly exist. Smaller structures are still useful and can be selected for. Even a naked bundle of photoreceptors can indicate to an organism which direction light is coming from. That's enough for it to move towards a region where it can do photosynthesis, or feed on photosynthetic microorganisms. The same can be said for metabolic pathways. They will claim that the Krebs cycle is irreducibly complex, but the evolution of this cycle is well understood, utilizing these same concepts of sequential changes and exaptation of existing proteins. They simply ignore the mountains of primary literature on these topics, knowing their targets will never read it either. A good analogy for irreducible complexity is complex ecosystems. These are comprised of many different species of animals, plants, fungi, and microorganisms. There is a delicate balance such that the removal of one species could cause the whole thing to collapse, just like one protein from the flagellum. But this doesn't mean it was poofed into existence precisely as it is today. Ecosystems respond to the stimuli that are present. Minor perturbations, slowly shifting inputs, and interdependence can emerge gradually. Tiny changes over long periods of time allow for a gradual response that produces a more robust system. A sudden dramatic change, like deleting an entire population of organisms, has a much bigger impact. That we can't start stripping away components of the ecosystem doesn't mean that simpler ecosystems can't exist, or that this specific ecosystem wasn't at one point much simpler. The same goes for any cellular component. In general, the pseudoscientific nature of intelligent design can be exposed simply by asking its proponents to put forward a rigorous model. Which biological structures are designed? Which are not? Is there an empirical way to test whether something is designed? Can they make predictions of any kind regarding biological systems? When asked, intelligent design proponents flat out admit that they can't answer these questions. They simply know design when they see it, which is a total cop-out. And also, they clearly don't, as we already showed with their most favorite example of the bacterial flagellum. Intelligent design is just a god of the gaps argument in a tuxedo, nothing more. Another aspect of genetics that creationists lie about is the proportion of the genome that is functional. Genes themselves make up around 1 to 2% of the genome. That is the percentage of actual coding DNA, whether protein coding or genes for ribosomes, transfer RNA, and so forth. Some time ago, it was thought that the rest served little to no function, and was therefore referred to as junk DNA. Over the decades, we have come to understand that there are sections that serve some role in influencing gene expression. There are promoters, enhancers, and other elements that have important functions. But this still puts the cap at around 10% in terms of the proportion of the genome that is functional. The rest remains junk DNA. This, of course, makes perfect sense in the context of evolution, as nature uses a large genetic canvas, and only certain portions of the genome evolve functionality by chance, while the rest must still be replicated and maintained at great energetic cost. Nature is not so efficient that it could have evolved genomes that are entirely functional, and the percentage of DNA that is functional decreases along with decreasing population sizes, since the greater the population, the greater the strength of natural selection on mutations. So many eukaryotes are able to accrue lots of junk DNA, as opposed to some bacterial species that comparatively have a larger proportion of functional DNA in their genomes. In the context of design, this does not make any sense. Why couldn't a designer simply design a genome without wasting so much space? Why include the 90% that doesn't do anything? Creationists will cite the earlier view that only genes were functional, vaguely reference more modern knowledge regarding sections with other functions, and say, see, there is no junk DNA. All of it has some purpose, just like the creator intended. But this is a fabrication. Most of the genome is indeed functionless. It is simply that a small fraction turned out to have functions that are now well understood by molecular biologists. Speaking of promoters and other genomic elements, plenty of research has been done surrounding these structures that specifically demonstrates how resourceful nature can be. Scientists have taken a wild-type strain of E. coli and stripped away the native promoter region of the lac operon, the gene necessary for metabolizing lactose. This is a segment of about 100 base pairs, which was replaced with totally random sequences, such that the gene will not be transcribed and the resulting lactose metabolizing enzyme cannot form. As a control, in one case, the promoter region was simply deleted and not replaced with anything. Needless to say, these bacteria could not survive on lactose alone because they could not metabolize it. Different clones with different random sequences were grown on a medium consisting of some glycerol, which they could metabolize, and increasing levels of lactose. After a few rounds of mutation and selection, clones arose which metabolized lactose. After about 100 rounds, these new E. coli clones thrived on lactose alone, and the newly evolved promoters functioned as well as the wild type, some of which were also paired with mutations in the gene itself, and thus different proteins. The only one that could never evolve was the one where the promoter region had simply been stripped away. This illustrates again the power of selection in vivo, showing how random sequences can acquire function in the presence of a selection mechanism, something creationists will forever deny, even when it is demonstrated for them clear as day. It also demonstrates another fact creationists always forget to mention, that there are many solutions to a problem, which means completely distinct proteins can perform the same function to a satisfactory extent, something which makes their big number games even more meaningless. Another creationist claim in the area of molecular biology pertains to soft tissue found in dinosaur bones. A T-Rex femur was found with the modified remains of flexible connective protein like collagen, fragmented blood vessels, and the remains of blood cells. This find was seemingly anomalous, as biological material was thought to not be able to survive for millions of years. Predictably, young Earth creationists claimed it was proof that this and every other dinosaur in history were not ancient but recent and walked the Earth alongside humans. But this interpretation ignores key facts and very obvious explanations. In reality, it is possible for soft tissue to be preserved under the right conditions. The process by which soft tissue decays depends on contact with air and water, but this soft tissue was in the dry interior of the specimen, stabilized by contact with hemoglobin and the mineral walls of the surrounding pores. Though fascinating, it does not change the dating of the specimen. In general, out of everything we could summarize regarding genetics and molecular biology and how these fields corroborate evolutionary principles far beyond reasonable doubt, we certainly can focus on mutations, embryonic development, and other such complex topics. But we also can simply point to heredity. This is something most people can wrap their heads around. Your genome is very similar to those of your parents, because it is a combination of the genomes of your parents. That's how paternity tests work, on the basis of genetic similarity. Using the principle of genetic similarity, we can tell who your father is, and your father's father, and his father before him, and so forth. So why would anyone presume that this concept doesn't work more broadly for genetic similarity between species? Humans and chimpanzees share 98.8% of their genome. This indicates very recent common ancestry. Other primates are slightly more dissimilar, so the divergence was slightly more distant, and we can do this type of analysis for any two species on the planet. There are so many other indicators that align with this principle. We already discussed orphan genes or de novo genes. When an organism develops an entirely new gene, that gene will continue to show up in its entire pool of descendants if the organism is reproductively successful. This is another marker of common descent. We can say the same thing for endogenous retroviruses, or ERVs. This is when a retrovirus, a special kind of virus that is capable of inserting its own genetic code into the genome of a host cell, does precisely this in a germline cell of an animal. Its offspring will then have that unique section of what was the viral genome, and this will be another marker we can use to identify common ancestry. There are countless ways to confirm evolutionary lineages that make no sense whatsoever in the context of design. We can expand this way of thinking to any genome as a whole. In order to discredit evolutionary theory, one would simply need to find two species that were anatomically so similar that they would have to be closely related under an evolutionary paradigm, but then discover that their genomes are such that they have a completely different suite of enzymes. This would be perfectly feasible if design were the case. Why wouldn't the designer experiment with different enzymes in similar forms? And it would be nearly inexplicable in an evolutionary paradigm, because how could so many enzymes become totally different in what would have to be relatively few generations? Of course, we never see this. The more related two species are on the tree of life, the greater the genetic similarity and the more enzymes they have in common. These are facts that do not disprove design, but when absolutely every aspect of biology conforms flawlessly to evolutionary principles, what room is left then for this God of the gaps that creationists would have you believe in? Not much room at all, in fact. And with that, we conclude the rigorously scientific portion of this series. Let's move forward and wrap things up with some miscellaneous arguments that creationists make that are somewhat more in the vein of philosophy or theology.